The overall goal of this methodology is to stabilize antibodies and proteins on a cellulose matrix to facilitate development of a naked-eye colorimetric immunoassay, which can be useful for point-of-care diagnostics.
This method can help answer key questions in the point-of-care diagnostic field. The main advantage of this technique is that it helps stabilize protein on cellulose, which allows the development of low-cost paper-based diagnostic assays. Demonstrating the procedure will be Gloria Chen, a student from my laboratory.
To begin, prepare one piece of square paper with a dimension of 1 centimeter by 1 centimeter. Also prepare 100 paper discs made from Whatman grade No. 1 cellulose paper with a diameter of 6.0 millimeters using a hole punch. To derive amino groups on the paper discs, mix 1 milliliter of APS and 10 milliliters of acetone in a 50 milliliter glass bottle in the fume hood. Add paper discs to the freshly prepared APS reagent mixture. Then, incubate the discs in the mixture for 5 hours with orbital stirring at room temperature. Decant excess solution from the 50 milliliter glass bottle into an organic waste container. Add 10 milliliters of acetone to the glass bottle, mix well, and decant completely to remove any unreacted APS and other impurities. Repeat this step 2 times. Spread the paper discs on the paper towel and place them in a 110 degrees Celsius oven for 3 hours. After allowing the paper discs to cool, store the discs in a 50 milliliter centrifuge tube at room temperature. Use Fourier transform.